Hello and welcome back. We are in part two now of the chapter on chemistry. We had finished up with lipids and we're transitioning to carbohydrates now. We're on to the second macromolecule, carbs. These are also organic molecules like the lipids were. And like the lipids, there's lots of carbon and hydrogen. However, there's also oxygen kind of thrown into the mix. That's what makes this thing a little more polar because the oxygen does not share the electrons evenly with carbons and hydrogens. And so that's how you end up with a partial charge and why carbohydrates are so hydrophilic, interact well with water. Let's go over to the name. Carbohydrate, carbon, and H2O. So the general formula is this: there's one carbon for every H2O in this molecule, and that's repeated some number of times. So for example, glucose is, this N is a 6. So C6H12O6. That's, again, carbon, hydrate. In case you ever have to come up with the formula, the carbohydrates are telling you what the formula is. All right, what they're for, in plants, for example, they can serve as long-term storage of chemical energy in the form of starch, as we'll see. In humans, long-term, like a day, a few hours, humans have we use lipids mostly as long-term energy storage, on our thighs and, wherever else we don't want it. But that's where we store our chemical energy for long term. So for a day or a few days, we can store energy in the form of carbs in our muscles and in our liver. It's certainly the ready energy store that we use in the blood. As we'll get to, these are carbohydrates. They are their own category. However, some small bits like simple sugars are also a part of these other organic molecules like nucleic acids. They can be converted to amino acids and be made into proteins. That's something the liver has to do for us. They can form a cell wall that humans don't have. We make cell membranes only, but the bacteria, they have cell membranes, and outside of that, they have the cell wall, largely made of something called peptidoglycan, which we'll see in a little bit more detail. And the animal cells interact with each other. There's this glycocalyx that helps the cells interact with each other. Okay, so that's like a laundry list of all these different functions of carbohydrates. However, let's talk a little bit more about the structure. Saccharide means sugar. So monosaccharide is going to tell you it is one sugar big. Disaccharide tells you that it is two sugars big. Both of these are referred to as simple sugars. Basically, they taste sweet to the tongue. We're going to call that a simple sugar. In contrast, polysaccharides, where you have hundreds of these repeating individual sugars, those do not taste sweet to the tongue. We call those complex carbohydrates, and we'll get to those. All right, let's do one at a time. We do monosaccharides. Here's glucose, the chemical formula for glucose. It actually makes a ring structure, and there's slightly different configurations of the glucose. And we'll talk about that when we get to talking about the polysaccharides. But for right now, all I want to show you is that this is a simple sugar. It's a single one monosaccharide. And here's a variation on this, the N-acetylglucosamine of NAG fame, NAG, of NAG and NAM, which are largely the blocks that are used for peptidoglycan in the cell wall of bacteria. Okay. The term disaccharide, as I've already said, it means two of the sugars hooked together. What we refer to as table sugar is this. It's two different monosaccharides, a glucose and a fructose. And they're put together when these chemically combine with each other. One of them loses this hydroxyl group, the other one loses the hydrogen, and that forms a water. This is often referred to as a dehydration reaction or dehydration synthesis because you're putting two things together. It's not just drying out, but chemically there is a water molecule being released. So that's where the word dehydration comes from. Anyway, this thing is called sucrose. And all of us have the enzyme needed to hydrolyze, which means to add water to. It doesn't mean just dissolve. I mean chemically adding water. That's the H goes onto one of the sugars and the OH goes onto the other sugar. And then you separate the glucose from the fructose. Okay, so that's a hydrolysis reaction. And lastly, the polysaccharides, where we have lots and lots of individual monosaccharides that have been hooked together into a larger structure. This is what plants use for a lot of their stems, and it's a structural molecule, cellulose. We humans can't digest this stuff. And it goes back to how the carbons are hooked together. Remember, we had, carbons are slightly different from each other. And you can have, or carbons, I keep saying that, glucoses are slightly different from each other. And yeah, you can have kind of alternating attachment structures. We don't have the enzyme that would allow us to break these apart from each other. So we humans cannot digest cellulose, but bacteria can. They do have the enzyme necessary for breaking this apart. The kind of polysaccharides that we can digest from plants are starches, and that the arrangement of the glucoses is a little bit different, but it's still a bunch of glucoses. And this is why even complex carbohydrates, even somebody who's watching their sugar, say because they're diabetic, you might think, well, they can eat complex carbs. No, it also increases the blood glucose because it's just a bunch of glucoses hooked together that then get separated and released into the bloodstream. Then we as humans, we do not store carbohydrates as starch. We store it in this slightly different arrangement called glycogen. It's a branching polysaccharide, and that's what we store in our livers and what we can store a little bit in our muscles. Okay, that is the end of part 2. See you over in part 3, where we will cover proteins.